I aim to run through 1.2.3 of the spec, covering the whole transport in plant section. Firstly, why do plants need transport systems? It's just the same old 1. Surface area to volume ratio and 2. Size. The rate of diffusion is not quick enough to supply cells deep inside the plant with the nutrients they need, but the dilemma unique to plants is that leaves cannot obtain water from the air and roots cannot absorb sugars from the soil, which is why plants have a transport system. This transport system is called the vascular tissue. The tissue, if we remember, is a group of similar cells that perform a particular function. If it's like a 2-mark definition here, it's important to say a group of similar cells, so you're implying different cells, particular functions, so they work together to perform something specific. Vascular tissue is made up by the xylem and phloem. The xylem and phloem are highly specialised, as I will go on to discuss. This is the distribution of xylem and phloem tissue in the leaves and the stem. I've left out the root, as I will cover that later, as it links in much better to the transport of water, which I think is quite tough. In the stem, the xylem and phloem tissue is found near the outer edge of the stem. The xylem and phloem together make up a vascular bundle. The vascular bundle is arranged as such in a ring, because this arrangement provides the strength and flexibility to withstand the bending forces to which stems and branches are exposed. Remember, the xylem is found on the inside and the phloem on the outside. In between is the cambium, a layer of meristem cells that divide to produce new xylem and phloem. I've added on diagram below the bundle cap because in the photomicrograph we can see this black thing. That's not the phloem. If you remember from when we looked at buttercups under the microscope, this is the bundle cap. Do not accidentally label it as the phloem in the exam. However, I don't think they'll stretch us that far. There is not much to remember about the xylem and phloem in the leaf. Main thing is the xylem is on top of the phloem. Once again, the xylem is on top of the phloem, and the vascular bundle forms the midrib and veins of the leaf. So let's move on to the phloem tissue then. Stem cells must first specialise to form the phloem tissue. Meristem tissue produces cells that elongate, and line up end to end to form a long tube. Their ends do not break down completely but form sieve plates between the cells. The sieve plates allow movement of materials up or down the tubes. Next to each sieve tube is a companion cell. Companion cells are very metabolically active, and as you will see, they're vital to the mechanism of translocation. Exam questions are always going to ask us to link structure to function, so here I go. 1. I'm going to start with the least convincing point. The sieve plates have lots of holes to allow solutes to pass. Clearly the holes are better than having dead end, but surely no plate would be best, like the xylem. It is exactly this uncertainty, which is one of the possible arguments against the mechanism of translocation, because the role of the sieve plate is unclear. And if evolution has taught us anything, things are there for a reason. 2. Sieve tube elements are lined up end to end to form the sieve tube. This contains very little cytoplasm, few cell organelles, and very surprisingly for a living cell, no nucleus, in order to not impede the flow of cell sap. Sugars are transported dissolved in water as sap. 3. The companion cell has a large nucleus to control the activities of the companion cell. 4. Plasmodesmata are fine strands of cytoplasm that link the protoplasm of adjacent plant cells through a thin area of cell wall. This allows for exchange of substances between the sieve tube element and the companion cell. 5. Companion cells are full of cell organelles, especially mitochondria, to produce ATP because the loading of assimilates into the companion cell is an active process. To split this point into 2 marks in the exam, say 1. Lots of mitochondria to produce ATP used for active loading, and 2. Lots of ribosomes to produce enzymes and carriers, for example the co-transporter proteins through which glucose is allowed into the phloem. Don't worry if you're slightly confused, I'm going to run through this mechanism of translocation in just a sec. On the right is a biological drawing of a transverse section of phloem tissue. The sieve tubes have very thin walls and are usually 5 or 6 sided, hence the hexagonal looking shapes. You can also see the sieve plates under the microscope. Remember, the same cell organelle can look different under microscopes due to the way the section has been sliced. Also there is some natural variation, just saying because I've seen this on a past paper and I got it wrong the first time. Translocation is the transport of assimilates, example sucrose or amino acids, throughout the plant in the phloem tissue. A source releases sucrose into the phloem, and a sink removes sucrose from the phloem. On this slide I keep saying protons because it's easier to type than H plus ions or hydrogen ions, but they're effectively the same thing. The most obvious source is the leaf, and the most obvious sink is the root. However, this can change depending on the time of year. So the roots could be a source in winter, but sink in the summer, for example. The slide outlines how sucrose enters the phloem, so I'll discuss the movement of sucrose along the phloem. There is a high hydrostatic pressure at the source because the sucrose in the sieve tube lowers the water potential, so water moves down the water potential gradient into the phloem by osmosis. Then at the other end, at the sink, sucrose is used in the cells surrounding the phloem. For example, sucrose may be converted to starch for storage. Starch is insoluble, which is why it is a better storage product than sucrose, as it won't mess with the water potential. Or the sucrose may be used in metabolic processes such as respiration. This reduces the sucrose concentration in these cells, so sucrose molecules move by diffusion from the sieve tube element into the surrounding cells. This increases the water potential in the sieve tube element, so water molecules move out by osmosis. Water moves down this hydrostatic pressure gradient and carries with it any assimilates. This is called mass flow, and depending on the conditions, the assimilates can move up or down the plant, and this is in contrast to in the xylem, where the xylem only transports water and dissolved minerals up the plant, only up the plant, not down. If you ring a tree, since the bark contains a phloem, the sugars can no longer be transported, so the bit above the part you've just cut off swells up. This is due to the accumulation of sugars, which in turn lower the water potential and cause water to accumulate too, hence further increasing the swelling. This tells us that sugars are transported in the phloem. An aphid feeding on a plant stem can also be used to show that the mouth parts are taking food from the phloem. And if you supply a plant with radioactive carbon dioxide, you'll soon find that the carbon appears in the phloem, which is once again another piece of evidence that this is the method for translocation. We know the mechanism requires lots of energy because the companion cells have lots of mitochondria, as previously discussed, and translocation can be stopped by using metabolic poison, which inhibits the formation of ATP. The rate of flow of sugars in the phloem is so high that energy must be needed to drive the flow. Diffusion alone would not be enough. This is the current accepted theory, as the pH of the companion cells is higher than that of the surrounding cells due to the pumping out of hydrogen ions. Remember, pH is a measure of the number of hydrogen ions. However, there is also some evidence against the mechanism. Not all the solutes in the phloem sap move at the same rate, whereas mass flow would suggest they should all move at the same rate. Also, sucrose is moved to all parts of the plant at the same rate rather than going to where it's needed most. And finally, as I touched on, the role of sieve plates is unclear. Thankfully, that's us done for the phloem, and now on to what I think is the slightly trickier stuff, the xylem and water. To produce xylem vessels, meristem cells produce small cells that elongate. The next step is then key. Their walls become reinforced and waterproof by deposits of lignin. This kills the cell contents. The ends of the cells break down so they become long continuous tubes with a wide lumen. Xylem tissue also helps to support the plant. The OCR definition for xylem is a plant transport tissue that carries water from the root to the rest of the plant. It consists of hollow columns of dead cells lined end to end and reinforced with lignin. It provides important support for the plant. On my little diagram on the right, I've tried to make it a point to highlight lignin, like we really need to remember this. The lignin thickening forms patterns in the cell wall. These may be spiral, annular, which means rings, or reticulate, which is a network of broken rings. If you look at plant tissue under a microscope and you see what looks like railway tracks, basically the straight lines I've tried to draw, you're observing the lignin thickenings, and that indicates you're looking at the xylem. So now I'm going to go for structure to function again. 1. Dead cells lined up end to end to form a continuous column, as the xylem wants to try and keep a continuous water column, which is important for the cohesion tension theory, which we're going to go on to. 2. Where lignification is not complete, pits in the walls allow water to move sideways from one vessel to another to maintain a continuous water column. This is really important for the cohesion tension theory to work. More detail in a minute. 3. The lignification kills the cell contents, and a xylem vessel has no end walls, no cell contents, no nucleus or cytoplasm in order to ensure a maximum rate of flow for the water. There's nothing to impede the water movement. So the lignin is not only key in killing the cell contents, which it does because it waterproofs the cell. The lignin strengthens the walls and prevents the vessel from collapsing. It helps keep the vessel from collapsing even when water is in short supply. And this sounds pretty analogous to the cartilage supporting our airways. 5. Lignin deposits also allow the xylem to stretch as the plant grows. And 6. The xylem vessels are narrow so that the water column does not break easily and capillary action can be effective. Going right back to basics, water moves by osmosis down a water potential gradient from a region of high water potential to a region of low water potential. Pure water has the highest possible water potential of 0 kilopascals, and everything else is more negative due to the addition of solutes. The scale is a bit weird. Now a bit of GCSE. Put an animal cell in pure water and it will burst. Put a plant cell in pure water and it doesn't burst because it has a cell wall. Once a cell is full of water, it is described as being turgid. The water inside the cell starts to exert a pressure on the cell wall called the turgor pressure. As the pressure potential builds up, it reduces the influx of water. However, if you put a cell in a highly concentrated solution, water moves out of the cell down a water potential gradient. An animal cell would shrink and appear wrinkled, we call it crenated, or a plant cell would become plasmolysed. Water loss in a plant cell causes the cytoplasm and permanent vacuole to shrink. Eventually the cytoplasm no longer pushes against the cell wall, and when the plasma membrane loses contact with the cell wall, we say the cell has been plasmolysed. I've brushed over this relatively quickly because it's pretty easy, but remember in an exam we always have to state the obvious because no prior knowledge is assumed. Now finally to the stuff on the slide. How does water move between cells? Firstly, to remember the apoplast pathway, try to think where the word has come from. Apo means away in Greek, and the word means away from the plasma membrane, i.e. water moves from cell to cell through the spaces in the cell wall, until of course it reaches the endodermis. Then it is forced to take the symplast pathway, which I know from German means through the cytoplasm. Water faces less resistance via the apoplast pathway, so if given a choice, would take this path by default as it is the quickest path. Since water travelling by the apoplast pathway does not have to pass through the plasma membrane, it can carry inorganic ions, plant hormones, proteins, sugars, etc., etc. And this is quite important, so remember that for the next slide, please. Now this is the distribution of the vascular bundle in the root. It is found in the centre, and there is a large central core of xylem, often in the shape of an X, and the phloem is found in the arms of the xylem. The vascular bundle has been arranged like this because it provides strength to withstand the pulling forces to which roots are exposed. Now I've held this back till now because it links in well to how water is taken up from the soil and into the xylem. I'm going to therefore take the opportunity just to talk around these pictures. So top right is a photomicrograph for the arrangement, and top left a copy of the diagram in our books. But if you look at that diagram along with the image I've scanned in from a CGP guide, it all makes a lot more sense, to me at least. Firstly, epidermis is a general term and just means the outer layer of cells of any multicellular organism. My first pitfall was getting confused between the role of the epidermis and the role of the endodermis. They sound very similar. The epidermis contains root hair cells that increase the surface area of the root and can go between soil particles to absorb more water. These root hair cells work by absorbing minerals by active transport using ATP for energy. The minerals lower the water potential of the cells and the water follows. This, as you'll see, becomes a bit of a recurring pattern, so we can remember these kind of like little patterns and it becomes easier to answer the question in the exam. So that's your first outer layer of the root cross-section. Then water moves through the cell cortex by the various pathways discussed on the previous slide. If you look at the root cross-section around the vascular bundle is a special sheet of cells called the endodermis. And inside this endodermis there is a layer of meristem cells called the pericycle. Remember, it's called the pericycle. I didn't really know where to fit this in on the diagram apart from obviously inside the endodermis. But maybe this is the kind of stupid thing they'll throw into our exam. It's the one word in the book that's only mentioned once, but somehow will become a discriminating question. The endodermis is also sometimes known as a starch sheath. The fact that it contains granules of starch should be ringing alarm bells that this is an active process. The endodermis consists of special cells that have a waterproof strip in some of their walls. This is the Casparian strip, and it blocks the apoplast pathway, which has been carrying lots of minerals and ions. By blocking the apoplast pathway between the cortex and xylem, this makes all the ions and solutes pass through the cell membranes. The cell membranes have transporter proteins, and the ions are actively transported into the xylem. This lowers the water potential in the xylem, so water from the cortex cells follows into the xylem by osmosis. Once water has entered the xylem, it cannot pass back into the cortex as the apoplast pathway of the endodermal cells is blocked. One way water moves up the xylem is simply by the root pressure that drives the water. However, this can only push the water up a few meters, yet trees can grow to way over 50 meters. The next two factors are transpiration pool and capillary action. To consider these, it is very important that we consider the structure of a water molecule. Its polar OH bond forms hydrogen bonds, and this is what gives water its cohesive and adhesive properties. Cohesion is the attraction of water molecules for one another, and adhesion is the attraction of water molecules to the walls of the xylem. So we get that the right way around. Cohesion is the attraction of water molecules for one another, and adhesion is the attraction of water molecules to the walls of the xylem. This is due to hydrogen bonding. So capillary action is where the water molecules are attracted to the sides of the xylem vessel, and because the xylem vessels are narrow, these forces of attraction can pull the water up the sides of the vessel. Before talking about the transpiration pool, let's quickly start talking about transpiration. The definition of transpiration is the water loss by evaporation from the aerial parts of a plant. Once again, transpiration is the loss of water by evaporation from the aerial parts of a plant. Water loss by transpiration is unavoidable because stomata are open to allow gaseous exchange, photosynthesis, which is a vital life function, and water can escape through these air spaces. With transpiration, remember that water must evaporate first before it's lost, and water vapour moves down a water vapour potential gradient. As water is lost, it must be replaced from below, and water moves from the xylem, from the roots, to replace the water lost. This is known as a transpiration stream. The transpiration stream creates a tension on the column of water. The xylem vessel is strengthened by lignin to prevent collapsing under this tension. Before moving on, I feel it's necessary, just for a sense of completeness, to mention why the movement of water up the stem is useful to the plant. Water is required in the leaves for photosynthesis. Water is required to enable cells to grow. Water keeps the cells turgid. The flow of water can carry useful minerals up the plant. Evaporation of water can keep the plant cool. So now to look at factors affecting the rate of transpiration. This slide is all about the environmental factors and revolves around some of the basic principles of GCSE physics. High temperature means molecules have more kinetic energy, so the rate of diffusion through the stomata increases, and so does the rate of evaporation. A higher rate of evaporation means there'll be a higher water vapor potential inside the leaf, and molecules will leave quicker, so the transpiration rate increases. Also, a higher temperature decreases the relative water vapor potential in the air outside the leaf, as molecules whiz around faster and further apart. This once again allows more rapid diffusion of molecules outside the leaf. If it's windy, then the wind will carry away any water molecules that have just diffused out of the leaf and maintain a steep water vapor potential gradient for a fast rate of diffusion. Alternatively, if the air is humid, there is a smaller water vapor potential gradient, so the rate of transpiration is lower. Finally, for the environmental factors, plants have some behavioural adaptations, so if there isn't much water around, the plant will try and close some of its stomata and less water will be lost. As well as environmental factors, you have plant factors. Plants are well adapted to reduce water loss so that they can survive in arid conditions. Plants well adapted to reduce water loss so that they can survive in arid conditions are called xerophytes. If absolutely stumped in the exam, don't panic. Think about these factors and think of the possible subsequent adaptations a plant could have. I'll try and show you how. So a plant with more leaves has a larger surface area over which water vapor can be lost. So think xerophyte, it's going to have smaller leaves, fewer leaves, or for example, needle-like leaves. A waxy cuticle is waterproof and therefore reduces evaporation from the leaf surface. So xerophytes have a thicker waxy cuticle to further reduce evaporation. The key here is to say a thicker waxy cuticle because plants have a waxy cuticle anyway, so just saying have a waxy cuticle won't really get you the mark. If the leaves have many large stomata, then water vapour is lost more quickly. Having stomata on the lower side of the leaf reduces transpiration. Also, closing the stomata when water availability is low will reduce water loss and so reduce the need to take up water. Other xerophytic adaptations you could easily remember are hairs on the leaves or rolled leaves. So this is all about basically trapping the water vapour, and you have a shallow water vapour potential gradient, so less water loss. One harder point, but probably worth remembering, is that xerophytes could have densely packed spongy mesophyll. This reduces the cell surface area that is exposed to air inside the leaves. Less water will evaporate into the leaf air spaces, reducing the rate of water loss. So for these questions, it will probably be 2 marks per point, 1 for stating the adaptation and 1 for an explanation. Last slide, not really going to say much, just we should really know this diagram pretty well by now. I hope the video has been useful. I've missed out talking about guard cells, and I've missed out totally about talking about petometer, but I've covered everything else in this part of the spec. Thanks for listening.